Our next speaker, the last for today, is Patterson. He is currently a assistant professor at Georgia State University in Atlanta. He obtained his PhD in SFU in Canada. He then traveled a bit in Europe for his postdocs. He spent a few years here as well in Milan, and now he has this position in the US. Um, he also has been working a lot on different topics in bioinformatics, and starting from more theoretical till now starting to work also with deep learning methods and machine learning methods. And today uh, it will tell us about characterizing viral subtypes for genomic big data. So thank you again, Mari, for accepting this invitation, and please, the stage is yours.
Okay, thanks very much, Simone. Yeah, so as Simone said, I did my PhD out in Vancouver, and then after that, I spent several years doing postdocs in Europe. And my PhD started out being quite theoretical, kind of combinatorial and approaches for many different things. But I kind of got into bioinformatics near the end of it, and then started doing kind of more applied stuff since I started in Europe, and now since there's so much biological data available these days, it's kind of really pushed it into the big data realm. So one cannot really escape machine learning in when that's happening, right? So indeed, the title of my talk is characterizing viral subtypes from genomic big data, and I'll kind of go through kind of a bunch of different work that me and my students really here at Georgia State have worked on over the last 3 or 4 years. So so yeah, uh, so I just wanted to um, I just wanted to kind of give this slide to kind of try and connect what I'm working on to pangenomics, since this is a pangenomics biohacking conference, and really it's one could view viral subtypes as a pangenome in some sense. So what this is here is a K-STEP network of a bunch of different uh, hepatitis A virus sequences that were taken from a bunch of different patients in the U.S. during the 2017 and 2018, and this was really gathered from the CDC here in Atlanta, and where Georgia State is situated, and what's interesting, so it's really they just took a bunch of sequences, and then they built this K-STEP network on it. So what it is sort of like sort of a neighborhood network. Like technically, what a K-STEP network is a union of all spanning trees of of a of a graph, and basically the graph is such that each node each haplotype, viral haplotype, is a node in this network, and then uh, two nodes are like a one-step neighbor from each other if the Hamming distance is one, and it's a two-step neighbor if the Hamming distance is two, and so forth. And so how you build a K-step network is that you first just take all the nodes disjointly, and then you build a one-step spanning tree on it, and if it's not a single connected component, then you join the two-step neighbors, and so forth until the graph is connected, right? So what that means is yeah, like you can see that this sort of bridge between the two kind of main subtypes. So you've got red subtypes and blue and a blue subtype. These are kind of two major clusters in this network. These ones are very far from each other. So what's interesting is yeah, like we really see like this was gathered from about a bunch of different patients, but we're really kind of seeing like two subtypes that kind of arisen, like obviously this sort of jump only happened in a couple of patients, uh, and then those patients were unfortunate to infect um, kind of another group of people, and this whole new subtype kind of broke out. And and the theory is that a lot of viruses are kind of happening that way. So I'm going to kind of look more like what I'm going to present is more like unfortunately like with SARS-CoV-2, I'm going to present SARS-CoV-2 kind of subtype, and unfortunately there we don't have much data on interhost heterogeneity, but the theory is that that's really what's happening even with SARS-CoV-2, but we don't really see it. So I'll be looking just at like analyzing SARS-CoV-2 just at the level of a population. So like you you're really looking at that you're viewing each person as having a unique just a single strain, basically the dominant strain that they contain. So but yeah, this is sort of interesting. But even looking at that, you can kind of see definitely close relationships between the different SARS-CoV-2 subtypes, right? So yeah, so so basically this interest in SARS-CoV-2, like maybe sort of motivated by this, the interest in SARS-CoV-2 was yeah was to have a look at it and try to do some clustering similar to this, but on the SARS-CoV-2 data that became quickly available as the pandemic evolved, right? So like what was interesting, I kind of started this position at GSU right at the beginning of the pandemic. So we were sort of doing research as the data was coming out. So it was quite that was pretty unique, I guess. Um, so we started looking at trying to cluster SARS-CoV-2, like the virus that causes COVID-19, um, according to entropy clustering. So we would really look at like you'd have to align the sequences, and then we're looking at a particular site, like this entropy clustering is all with respect to just a single site, and we're basically trying to like cluster the sequences in such a way that the overall entropy is the lowest, right? So you'd kind of like the way you would view this is maybe like every site that has nucleotide A is sort of in one group. Like suppose you had four groups, right? You could just cluster perfectly the at this site, right? So like everything with state A would be in one cluster, everything with state C would be in another cluster, and so forth, right? And so, but of course, you're trying to do this globally over all sites, trying to reduce the site-wise entropy. Like you're trying to minimize the site-wise entropy over the entire sequence in trying to do this, right? And so it's sort of an interesting, like it's sort of like a passive way to do clustering in some sense, because you're not trying to like forcibly try to optimize. Like you are trying to optimize this, but and actually the theory is that I guess it is a convex function, so you can optimize it. That's another thing we tried, but just looking at this as a measure of sort of cluster quality, we kind of this is kind of how I started looking at or we I guess started looking at the clustering SARS-CoV-2, and there's many other sort of clustering measures as well. Actually, there's hundreds of them, like the Kalinsky, Harabaz, and Davies Bolden index, and there's so many, right? Um, and so yeah, and then of course like yeah, so this is just the site-wise entropy, and then of course you want to look at you want to look at this would just be the entropy of a cluster, but then if we want to look at a clustering, this would be the entropy of a clustering, so of of sequences. And so we took this idea and ran with it with current GSAID data. Like so, GSAID is a database that I think most of hopefully most of you have heard of that. and it's so here we had about 1 million sequences, and we did this kind of clustering, and of course we did different, like we had to represent the sequences somehow, so we did a simple, like I guess this, like implicitly there's some sort of encoding that we're of the sequences, some sort of numerical encoding that we've I mean, here it's really just like the sequence itself, so there's there could be four nucleotides and a gap symbol in each site. um, and yeah, so yeah, so here we ran like various approach, and this was, yeah, so we tried different ways of performing clustering. uh I think we tried k-means and yeah, several other, but what we found out was that k modes was the best way to cluster the sequences, and like, so that's really the approach. That's like, so yeah, we're applying k modes to the sequences. So I think one can imagine how that would work, right? Like the each like you're trying to do it at each site, right? Like if you think about how k modes works, it's building these clusters, and adjusting it based on the mode, where the mode is sort of like the most dominant nucleotide in the cluster at the moment, right? That would be the mode of a cluster at a given site, right? And so you're trying to run all of this with k modes, and then we do different ways of trying to initialize using different types of distances, like Hamming distance or this TN93 distance. I won't really get into this so much. This is kind of a work of another group, Clique SNV, that kind of identifies viral haplotypes, so it can be viewed as a clustering method as well. And uh, so we took that approach, and then to sort of validate it experimentally, we ran it on this. I think it was, we call it the UK data set, but I think it was the Embl Ebi. It was a data set from Embl Ebi in the UK, right at the advent of the alpha variant in the UK. Like that's really where it started, and we performed this clustering. So this is sort of just a distribution of all the clusters, and we never labeled these clusters, but of course at the end we noticed this one cluster. So what's interesting about this sort of clustering approach is that it's like we do clustering sort of, like, I guess I say this in some of the talks, but it's an alternative to phylogenetics methods. But in some sense, we're kind of trying to tackle a different problem. Like we don't, it's maybe where, like, it can be faster than, it certainly is faster than phylogenetics, but now I think the SARS-CoV-2 phylogeny is fairly well resolved, and so you can do these phylogenetic placement methods to kind of perform clustering of new subtypes that come out. But if a new was to happen, for example, one could do this more primitive kind of clustering approach to kind of really understand what clusters are growing quickly in the population versus others, right? And that's kind of what we did, because this was at the beginning of the SARS-CoV-2 pandemic, and the first major subtype was alpha, and here we're using the clustering method just to kind of see how the clusters grow as time goes on, right? And indeed, we were able to pick up this alpha variant, which quickly grew at the end of the year in 2021. This is, or no, I'm sorry, 2020, I suppose. So yeah, so that's just sort of experimental validation of this. So with this sort of work, we sort of ran with this clustering idea and produced a whole bunch of different approaches basically to cluster, and we, like, this really didn't make use of machine learning per se. I suppose like k means and k modes could be viewed as that, but it was sort of a clustering, yeah, just to see if we could do clustering to identify subtypes of SARS-CoV-2. That was really the point of this work. And So we really ran with that idea, and then uh, my basically my students kind of ran with that idea, I suppose, and we really we started getting into like Sarvan Ali had joined recently, right after I had done this previous work, and he's an expert in sort of these embedding methods and applying machine learning, like kind of embedding methods that represent the sequence, that represent the sequence well, um, so that machine learning approaches can be used for clustering or classification. And so this is just a clustering approach, but it's sort of a proof of concept, kind of the first proof of concept of machine learning being applied to the sequences. So I'll kind of zip through the motivation. So it's really trying to trying to understand the the diversity of variants. I guess like, just to be careful, the I mean, this is kind of how viruses evolve, but SARS-CoV-2 actually evolved very conservatively. So there's actually, like, it's all the same strain technically from a virologist's point of view, and there's just variants in there. So it's really like, the, despite the fact that it evolved in a very conservative way, there was quite a few variants, and they definitely have different effects, which is quite interesting of SARS-CoV-2. So maybe that even connects it more to this idea of pangenomics, that we have like, I don't think we've ever had so many sequences in the history of sequencing. Like, there's 20 million sequences of SARS-CoV-2 available in databases such as GSAID. Like, right now we had several million, but now there's 20 million. Um, and it Yeah, so never, I think never in the history have we sequenced one virus, and it's not very diverse compared to, like, even this hepatitis, for example, or HIV. But the data is here, so we took advantage of it with different methodologies, and so a little bit about the SARS-CoV-2 genome. Like, what we did in a lot of studies was just look at the spike protein. So because the data was so large, we were looking really at the spike protein, because mutations are sort of happening, uh, let's say, disproportionately in that region, because it's the region that's important for really attaching to the human, the host cell membrane. I shouldn't say human, because there's been spillover, like zoonotic spillover of SARS-CoV-2, and so and so applications of this are sort of, I hope I sort of motivated the application already. We're looking at sort of how the subtypes, if a certain subtype is growing very quickly, this kind of primitive clustering method can quickly identify which one is, and one could direct efforts, like sort of lockdown efforts, or even vaccine design efforts. Certainly, vaccines have come out for certain variants in the last couple of years. Yeah. So here what we really wanted to do was it's really focused on the embedding method now. So before, the embedding was we just took the sequences directly and performed k-means and k-modes on such sequences. But we in some sense the embedding was really a one-hot encoding, one could view it, because we aligned all of the sequences, and each, in some sense, we perform that, right? So we still did, like, somehow, like a fixed length feature embedding, right? And then applied k-means or k-modes. So here we do something slightly different. So we tried it. We proposed a method based on k-mers, and so, like, hopefully most of us know what a k-mer is. A k-mer is just like a contiguous substring of the string, a contiguous substring of length k, right? And so So if we had this sequence, for example, that there would be three thremers, like we you can kind of build this representation by just sliding a window over the sequence, right? So the first streamer is ACG, the next streamer is CGU, and the third streamer is GUA. There would be two formers and one fivemer, right? So this is sort of the idea. So we're gonna generate um, basically from this what we can do is build a frequency vector of a sequence. So what's interesting is this frequency vector grows with k rather than the length of the sequence, and so it's a good way to build a compact um, vector represent like, and it's a fixed length representation too, right? Because it would be like its length would be the size of the alphabet to the power of k, right? Or it's the other way around. Um, no, it would be that, yeah, the size of the alphabet to the power of k, and so with this representation, we took about yes 60,000 sequences, and this is the data we had at the time, and then performed yeah, so this is really the detail. So we generated frequency vectors based on k equals 3, and so we just chose three based on we tried different values of k, and found that three was the best for this data set. And yeah, and then we just applied k means on the frequency vectors. And so and with that, like, since we were performing clustering, like, it was sort of, I mean, in the parlance of machine learning, it's a unsupervised learning. We had to see what the optimal k was for this, right? So just a bit on the 60,000 sequences. We did a bit of visualization to understand how the data was structured as well. In some senses, maybe this TSNE, so the T-distributed stochastic neighbor embedding is sort of a way to embed a high dimensional data set into sort of two dimensions. Well, it's into a low dimensional. I mean, like, TSNE is more general than just two dimensions, but the typical use case is sort of two dimensions, and it tries to do it in a way that neighborhoods are not disrupted too much, right? So things that were close to each other in the high dimensional space are kind of close to each other in the low dimensional space. And so, and what I mean by a high dimensional space in this case is that each site in the aligned sequence, or in this case, each kind of column of this fixed length embedding, like, all of these are taking some sort of fixed length embedding. In our case, the fixed length embedding is really there's sort of a column in this embedding for each k-mer, and the frequency of the k-mer is basically the number at that column, right? And so we we perform this to kind of understand how the embedding is representing the data in some sense. Like, is there a natural clustering kind of arising there? And so and then to really the results of this. So this was just a clustering method, so we didn't really have a ground truth to go on, but what we did was just use we labeled the cluster with the variant that is the majority in that cluster. So by looking sort of the F1 score, like that would be the ground truth sort of labels of these clusters. And then if you look at the F1 score of this, you get an idea of, it gives you an idea of how maybe pure the clusters were when generated by these methods, right? So we tried these different approaches with sort of like just pure k means with a ridge regression, and with lasso regression. So the details of these I'm not completely um, sure of, but yeah. So this is sort of the idea. So it was really sort of, this is the first time we tried Kamers to perform to perform sort of like a Kamer-based embedding to perform clustering of the sequences, right? And so I think yeah, I can, and we track the variance, we track the spread of the variance over time with this, and it seemed to sort of agree with common, yeah, with common. You can see that one variant goes out, and that's sort of how the evolution of the variants have occurred with SARS-CoV-2. So like one would die out and a new one would rise, and so forth, right? So yeah, and this happened with different degrees of intensity in different countries. Like for example, the alpha variant had sort of a brief life in the U.S. compared to India, for example, where there's many more flights to and from the UK to India than there is in the US, for example. And yeah, so we also did, yeah, I did want to mention this as well. I mean, this is maybe a bit of a bold statement, but we did look at information gain of the different kind of in this case, we were really looking at, again, a one-hot encoding, because we really wanted each position in the embedding to represent the actual amino acid. So this would be really a position in the aligned sequence. Um, so here we looked at information gain, basically like how much information does a particular amino acid position give to the clustering, right? So is it important in deciding whether a variant is an alpha or a gamma, for example, or not? And we did like we did look at some of these high information ones, and they seem to correspond to sort of the mutations that characterize, for example, the alpha variant versus the wild type strain, for example. So as sort of a validation of this approach. And so yeah, so we took that idea, the same idea with the Kamers, and ran with it to get a really scalable one as data started coming out. So this was sort of the paper that launched Sarvan's PhD thesis, I guess. So it was really a proof of concept that really proof of concept that we can do these embeddings and really scale to millions of sequences. So here we scaled to 2 million sequences, and it was quite interesting because um, because at that level you're getting really good accuracies because you have so much data with this sort of, so it's really sort of the proof of concept of using machine learning for characterizing viral subtypes, right? And yeah, so yeah, like to go through, like, yeah, briefly go through this sort of intro. I mean, we're sort of understanding this now, like yeah, indeed, like the data that kept coming out was really pushing genomics into a big data realm. So can we use these sort of methods for it? And yeah, and really the problem was, can we design a representation that allows to scale to millions of sequences? And so again the the approach was really was again just Kamers, right? So we use Kamers, and then a few other techniques to kind of reduce the dimensionality of that representation, and and it really worked out, right? So yeah, so indeed, like the Kamer, the length of this representation is the alphabet size to the power of k. Like you're sort of, it's a trade-off between a longer embedding and how well you're representing the sequences. You have to find out the proper trade-off. we went with k equals 3. We found that that was a good trade-off in this case. But of course, like at this level with 2 million sequences, it was actually 2.5 million sequences. You have to do a few other tricks. So like the representation is not compact enough to scale to that level, right? And so we did the, I guess the main, like we tried a few dimensionality reduction techniques. I think ridge regression and uh, but the big one was this random Fourier features method. So what it was is like one way to kind of speed up computations is that you're like a lot of these methods like support vector machine or even k-means is really clustering based on pairwise comparisons. And so one way to speed things up is to build sort of a kernel matrix which stores these pairs, and then you can access you can access really quickly. So like if this was this distance, if f was this distance, um, uh, one can do it, right? And then there's this all of this, like I'm not an expert on kernel matrices by any means, but the the theory says that any positive definite function can be represented with a dot product. Um, but the problem with this kernel is like if you're really trying to run comparisons with like if you're trying to perform this on 2 million sequences, uh, there's no way you can store like a 2 million by 2 million matrix, right? You still have to store all pairs. And so this is a different paper that came out in the early 2000s, I think, that comes up with a kind of a low dimensional approximation of this kernel matrix. And so one can now just access this with a dot product, right? So basically what we're doing is we're building the embedding and then casting it to this lower dimensional embedding, and then running the downstream methods on that, right? Like k-means or k-modes and so forth, right? So that was the basic approach. Like RFF was really the one that allowed us to scale to that level, right? So we so just by stitching some things together in a clever way, we we were able to get something to work. So we and this was also the first paper where we performed classification. So the GSAID database comes with like there's 20 million sequences there now. At the time there were several million when this paper came out, and each sequence is also labeled with some metadata, and one of the metadata is his lineage label. Like for example, alpha is B117, and this lineage itself is parts of it are manually annotated, but parts of it are also automatically annotated. So it's a little bit of a chicken and egg, or maybe dog chasing its own tail sort of issue, because we're using a machine learning to kind of validate another machine learning approach. But in some sense, the development of methods is still important in this case. So what we did was we use these ground truth labels that are in the GSAID database for the purposes of classification. So there we have these ground truth labels, and then we can run classifiers and determine accuracy, recall, etc. Right? And so in this case, we really looked at 2.5 million sequences, and it's obviously dominated by the alpha variant because that was the one that was big at the time. So there's some data imbalance issues there. I guess the delta and the alpha were these are both of these uh, subtypes there, and and ran with this method, right? So again, yeah, I think I've already explained sort of this, and so here we like we called this spike DVEC, but really what it was is we took the spike protein, computed the Kamer representation, and then performed RFF. Like we basically took the spike protein, built the frequency vector, the Kamer frequency vector of this, the, and then we performed this RFF to get this lower dimensional embedding, this much more compact embedding, and then ran yeah, different machine learning approaches on it, right? Naive Bayes lasso regression and ridge classifier, and so and so that's what spike DVEC is, and this is what the one hot encoding. So that this is what a straight up one hot encoding would do, right? Like the one that I presented at the very beginning, for example, where I just take the sequences and I align them, and then I just look at the different nucleotides. Like I guess technically a one hot encoding is a binary encoding, so you're building these vectors of ones and zeros based on like a binary representation of these aligned sequences. But so I would have like if I had four nucleotides and a gap symbol, it would be like 0, 0, 0, 1, 1, 1, and 1, 0. You know what I mean? Like so at a particular representing a particular site, and yeah, and so the so this is sort of a proof of concept that the embedding matters, right? Because this would be this one hot encoding was basically what we did in the first paper with the entropy clustering that already performed like fairly okay, um, but the embedding matters, right? So like these kind of more like compact embeddings that really capture the information properly or like in a compact way can really boost both the runtimes and the accuracy, right? And so this sort of launched like we have tens of papers kind of based on this where we performed like yeah like a whole bunch of different types of embeddings on many different things. We also looked at a bunch of kind of feature selection methods as well. Um, yeah, and really sort of explored this use of machine learning for the classification of these SARS-CoV-2 variants, right? Because we had this like the data was growing and we kept running with that, so it was a fruitful time. Um, and this was the clustering results. I guess I sort of talked about that in the other paper, so I won't go um, too much in that detail. And I already talked about feature importance. Um, so indeed, Spike DVEC kind of launched a whole set of sort of really what it was embedding approaches for like kind of like in machine learning parlance, like feature engineering, right? So handcrafted features in such a way that you pass it to some machine learning approach, and and it learns the labels from this embedding, right? And so if the embedding kind of represents the sequence in a nice way, then we're getting good results, right? So I see that I have about 5 minutes left, so I might speed through. Um, I might. So we tried like Spike DVEC definitely proved that concept, and we tried a bunch of different machine learning approaches, um, and and then we tried some different things like I'll quickly go through here, I guess, uh, like here we were trying to we were predicting subtypes or like learning subtypes from the data. Here we were looking at like a different type of metadata, the country that it came from. Like can we sort of characterize uh, certain sequences based on the country they're in, right? Because like definitely waves don't happen uniformly around the globe, And so that was sort of the idea. Same data set actually. It was 2.5 million sequences, and and same sort of methods, but a different sort of approach there. So we were really trying to understand like can we identify what country we're in based on a collection of sequences. Um, yeah. Uh, and oh, so we try kind of straight up uh, like let's say Euclidean embeddings in Euclidean space for trying this. So we we started exploring maybe slightly different embeddings because like really like one thing about and this is another student who's in my group like Sarvan and Prakash are kind of the two senior students. Sarvan actually graduated recently, and he's at Columbia now. Actually, he got a postdoc in Columbia, so that's great. And Prakash is near the end of his, so it's a we worked we worked together on uh, non-Euclidean embeddings, right? Because the sequences like Euclidean embeddings, I think we kind of proved that they work pretty well for this kind of classification business. But in reality, there's like in reality the sequences are sort of like they're related to each other hierarchically, right? There's sort of a hierarchical relationship between the sequences, right? So we tried to kind of explore that. Is that a more natural embedding? So really the idea is like I'll zip to the Poincaré disk maybe. So the idea is that the Poincaré disk I can really like it more naturally embeds a tree because I can put the root at the center and then the levels of children are like if they're really on this disk, this hyperbolic disk, there's less distortion in the space. It more kind of honestly represents the sequences, right? And so we kind of ran with this as an embedding method, and but we only tested it on about 7,000 sequences. So it's yeah what we call the Spike 7K data set. But it was a slightly different approach. Like not unfortunately not as scalable as the or at least we never tried as the Euclidean approaches because there's really these methods like k-means that are really optimized for these spaces, right? And then so I'm almost at time now. Maybe I'll go over uh, a little bit and talk a little bit about deep learning approaches. So these are all in the domain of feature engineering methods. But let's look at deep learning where we're not engineering features at all, but we're just letting the methods learn. So yeah. This was another actually this was like so like kind of Sarvan here was one of my first students who kind of started this, and this is his former supervisor in Pakistan, and he joined the group kind of at the beginning, and really like Spike DeVec was kind of what launched his career, and then another student Tasleen, this was sort of her work, so she really went into this sort of deep learning approaches based on chaos game representation. And so this was one of our first papers exploring kind of deep learning approaches to to this domain, right? And so I won't go too much in that. I mean, like the application is still kind of the same, uh, and so really like so the chaos game representation, just to kind of just to try to understand it, like the first one that kind of came out was for nucleotide sequences where we like for example it's a way to convert an image into a sequence into an image, and so like each of the like when I have a nucleotide sequence is very natural because I can just have I can have four quadrants, right? One quadrant for each nucleotide, and then it's sort of recursive, right? So if I have GTT, it means that I like first I go to the upper quadrant, then I go to the lower quadrant, and then I go to the lower quadrant of that, right? That's why GTT is over here, right? So I'm really generating like, and this would really be a pixel. So I would light up this pixel if I had that if I had that sequence there, right? And so one could generate like yeah, like a Kamer based representation, and you would really get an image, right? So like if like in this case, like if I had tremors, for example, I would have the frequency of that pixel would be how like the hue of that pixel would represent the frequency of that Kamer in the embedding, right? And so that's all fine for uh, nucleotide sequences, but we were looking at amino acid sequences, like the spike sequence, right? So we had a slightly more generalized one where we really had this like kind of flakes, and so now you're really like embedding amino acid kind of on this ring sort of, and then these rings are sort of recursive. So you can see that there's a sub ring for M. So if I had like MM, then I would embed like at the top part of the sub ring, right? For example. And so we're sort of building an image that way, and and then yeah, and then the embedding, like I guess there's a lot of like we have a lot of other papers where we were trying like Kamers and minimizers and stuff like that to build different kind of feature engineering methods, and we were still doing those to kind of build this image. Like we were kind of we needed to still build a fixed length numerical representation and convert that into an image, um, and so we were certainly doing that. Um, this was another method where we were using, like this was another separate paper using position weight matrices to build a representation. But basically, like we were using all of these to these embeddings, these different kind of feature engineering embeddings to build images, right? So this was just a straight up like the straightforward chaos game approach, which is really just the generalization of this to an image, and then we were building different embeddings and converting those into images, and then we ran so we ended up with yeah, with five different representations, and then of course we would run a like we would really run like a convolutional neural net on this representation, right? So yeah, so that was really kind of the first kind of approach of deep learning on this data, and which is certainly appropriate since there's like tens of millions of sequences now. Maybe the deep learning methods can really extract some structure that other methods cannot. On the other side, I think that for that reason, there's a dire need for explainability. So like indeed these methods are performing well, but why are they performing well? What are they picking up that the feature engineering methods are not picking up? We certainly need to look more into that, right? Um, I'll go I'll go to one more. So again, this was again Taslim's work. She was really um, uh, she kind of did her thesis on she graduated now too, actually, and she's in St. Louis, and she really did her thesis on these uh, image based representations of sequences. So yeah, so it was quite nice. So this was another one where we built these kind of kaleidoscopic images of the sequences. Um, so so in some sense, yeah, it was again kind of a recursively defined recursively. So each amino acid would have a unique position, and then you're recursively building an image based on the sequence. So if I had A, C, like this would represent A, this would represent C. If then I had a Q, it's building this inside, and yeah, and then again we apply some yeah we apply some CNN downstream on these images for classification. Again, the input is really just the embedding and the labels, and we're learning. The deep learning method is really learning this embedding from the, or learning the label from the embedding. And yeah, and so I'm pretty much out of time, I guess. So I'll leave it at that. But hopefully I've kind of given you a flavor of different different sort of machine learning approaches that you can apply to sequences. So yeah. So I guess that's it. So thanks.
Thank you very much, Murray. Well, it was a very nice talk
Thank you.
and really dense, but at
Mm-hmm.
least we saw different parts of your work. Let me check if we have some question from the audience. I think we do not. We are running a little bit late, so I would just like to ask very quick one, which is regarding the first part of your talk when you were discussing about the phylogenetic analysis of viruses, or COVID specifically. Um, Maybe I missed that, but um, is the something like geographical position, like GPS coordinates, part of the clusterization method? Is it is it combined? If it's there, if it is it combined with genomic information, and if it's not, is there a way to combine them? What do you think?
Yeah, that's an interesting point. We actually never really I talked about like spike Spike was just straight up variants. It didn't matter. Like that was the only metadata that we were using, just the variant, like the lineage label. And then we also had this country one, but we never actually tried a combination of them. It's sort of interesting. One could like I guess you're sort of delving into this phylogeography domain, which I mean, there's books written on that. So certainly that would be like something interesting to look at. So the trees, like I mean, if you go onto NextTrain, like I think if I just like pop onto NextTrain, they certainly yeah, they certainly build like SARS-CoV-2 phylogenies. I think I can quickly okay, so maybe I can't find one right away there, but they certainly I'm sure they're taking into account both geography um, and sequence. We never did, but that's certainly something interesting.
Yeah,
There's
maybe.
there's way more to explore here, so it's
Yeah, maybe they could also be combined in into the classification aspect rather than the phylogenetic one.
yeah,
Uh,
certainly, yeah. That you could boost classification if you considered like it'd be sort of a multi-class
mhm.
classification. You could combine all of this possibly with would you even combine all of this with like a different embedding method where like locality is in some sense, yeah. I mean, the virus is sort of like evolving locally. I mean, like when it gets introduced into a new place, it sort of evolves locally. I mean, like it's kind of hard to, I guess it's kind of hard to track because I mean transport networks are so erratic. But yeah.
Yeah, so we have one question from the audience. Um, this is regarding the last approach. So this fractal-like structure that you have, kaleidoscopic structure, right?
Hmm.
Um, Davide is asking if you can give like a short list or a hint of possible uses of that approach.
Hint of possible uses of that. I mean, I guess like you wouldn't. It doesn't necessarily just have to be like I guess it's a way to convert sequences into an image, right? So it's but it really could be any sequence, I guess. Like it's really just converting like really just converting a sequence into an. So you could apply it to I mean, you could possibly apply it to paragraphs of written text and do sentiment analysis on it. Why not?
Yeah,
Like it's
so we can think of it like an extension of uh, of a Chaos game representation in a different way, where we can encode more a larger alphabet still in a 2D image, right?
oh yeah. You could, I suppose you could, right? Like I mean, you would lose sort of like if the alphabet got arbitrarily large, you kind of would lose some kind of like representation, like
Yeah,
capacity to represent that, right? Like here we have an alphabet of 20 with 20 unique coordinates. You could have an alphabet of size 100 with 100 unique coordinates, right? But the image would start getting. I'm sure the deep learning wouldn't be able to predict as well.
yeah, yeah, of
You
course.
move things closer to each other. Yeah, yeah, yeah,
Okay, great.
yeah.
Uh, I do not see any more questions, so I'll thank you again, Mari, for being here,
Excellent. Yeah.
and thank you also for all the audience. We are done for today. We will resume tomorrow at 9. Thank you very much, everybody, and have a nice
Thanks.
evening. Bye, Mari.
Have a good day.
You too.